This is a production of Cornell University.
It's an honor to be here, and it's a special honor to be invited by students. That's my first student-invited seminar, and I think you all know that that's a high plank to take. So thank you all for choosing me to come here, and moreover, I think like students here are so driven and setting such a high plank that they asked me to give 2 seminars and told me we're going to zoom in to see both of them. So I've ended up essentially composing 2 talks. And so if I'm not going to talk about some of the aspects of the research and you're still interested about it, just feel free to talk to me about it. And so so yesterday I spoke in Geneva and spoke more about some of our applied work and wheat and some of our functional work. And today I've decided to focus on what we're doing in the evolution of the plant immune system. And as was mentioned, I just recently moved my group essentially back home to my home department from England, where we've been before for about 4 years. And I'm quite excited to be back in the US and meet the colleagues here and establish more of a connection with US colleagues. And if I want you to take back maybe one thought from this, it is that right now is a good time to look at the plant genomics and look at the evolutions. And specifically what we try to do is to decode information in the plant genomes. And by that, I don't mean sequence and annotate. By that, I mean ask specific biological questions, hypothesis-driven questions that we can answer with the genomics. And if we understand the rules, how the genomes evolve and what's encoded in there, we hope that we can engineer the systems. What's been driving my interest in science for a long time is basically what is the difference between a diseased plant and a healthy plant. And as a whole lab, we think about it in 2 ways. One is a diseased plant that has a disease has to be a host, has to accommodate the pathogen and provide whatever the resources and nutrients that it needs. And at the same time, the pathogen needs to be able to suppress immune system. And a healthy plant can be either a non-host in a way that it just doesn't provide the right environment for the pathogen, doesn't have the correct susceptibility components, or you can have an active immune system that recognizes the presence of the pathogens, elicits the immune response. And then what I'm going to speak today is about that latter part about the plant immune system. And the big biological question is, how can plant evolve new pathogen recognition specificities? How does that system evolve? And here I want to bring back the Red Queen. And if you all remember, that's the moment in the Lewis Carroll where Alice meets the Queen, and the Queen says, well, in this place you have to run just twice as fast to get somewhere else, and you need to run to keep in the same place. And of course, that concept has been applied repeatedly to coevolution, and especially coevolution of plant pathogen interactions. And the reason why I'm bringing it, because when I was the first year graduate student, I happened to read this review where Stephen Hedrick from UC San Diego, he applied that metaphor to mammalian immunity. And he made a case of, even with the most advanced immune system that we have, where we can recognize literally any epitope, right, anything, we still get disease. We still get the common cold every year. So the pathogens, no matter how advanced the immune system is, the pathogens will still be able to overcome it. And we still need to keep up. And what about the plants, right? So the plant's life cycle is much greater than microbes. They don't have specialized immune cells. So how do they stay ahead, and how do they engage in that battle? And we think about the immune system from the perspective that the most important part in this is the generation of a diversity, which upon the pathogen infection provides enough material for select beneficial variants and then amplify them. Now, of course, in crops, a lot of times that's the very first level that's been lost. If we plant monoculture, we're losing that diversity, the first step. And the two approaches, though, my lab does both wet lab and bioinformatic research, and I'm guilty of being both of these guys. On the wet lab side, right, we study particular pathways, we study very specific proteins, and we still don't know, we still don't know exactly how plant immune signaling happens. We still have this black box. And at the same time, this is also us, it's also my lab. We can look at all of the data, and we can generate all of the answers at the same time. So how, and I think like the main outstanding question right now for all of you, like for you like students out there, how do we bridge that gap? How do we reconcile the bioinformatics of big data analysis with the mechanistic work of the pathway? And I think it's evolution. So if we look at it in the context of evolution, we can answer some of these questions. So what do we know? Before I go into the plant immune system, I want to quickly summarize the last 35 years of research in respect to the plant cell. And what we know today is that in plant cell, there are two major systems of monitoring the environment and responding to the pathogens specifically. One of them is receptor-like kinases or pattern recognition receptors that monitor the environment outside of the cell. And we all know that no matter which kingdom pathogens come from, they all shed molecules that reveal their presence. They're specialized to these pathogens, either it's a flagellin or it's a chitin. And that recognition signals the first level of immunity. What we also know is the pathogens from all of these kingdoms, they evolved abilities to inject specialized effector proteins within the cell. The mechanisms how they do it are different, but all of them do that. And those pathogen-derived effectors, they have two roles. They have the role in suppressing this immunity, and they also have a role in modifying the environment to enable the infection, to make the plant cell susceptible and the plant susceptible for the infection. And while this first role of the effectors in suppressing the immunity has been well studied, the second role of the effector modifications of the plant metabolism on the plant cell is much less studied. And those effectors, they reveal the presence of the pathogen now inside the cell. And the recognition of those effectors is mediated by intracellular receptors that we really like to study. They're called NLRs, or nucleotide binding leucine rich repeat proteins. And they can recognize the effectors in two different mechanisms. One of them is direct recognition, meaning like a ligand receptor interaction between a pathogen-derived molecule and an NLR. Evolutionary, if you think about it, that's not very efficient. Because plants, I told you, they have an innate immune system, they have a limited number of receptors. And using these receptors to monitor all of the effectors from all of the pathogens evolving all of the time, that's just not realistic. So what plants evolved, they evolved a second mechanism, which is similar to the first one. But instead of monitoring the effectors directly, they monitor their own plant proteins. So instead of having recognition of non-self, they have a recognition of modified self. And that is, right away, that's a much more limited number of proteins, right? And moreover, what's known is that different pathogen effectors, they, again and again, they target the same plant proteins. Because at the end, different pathogens, they want pretty much the same things from the plant. They want to get the nutrients, they want to suppress the immunity. So what ends up that by monitoring the plant proteins that are modified by the pathogens and monitoring specifically those hubs that can be modified by multiple pathogens, plants gain evolutionarily the ability to recognize different effectors and different pathogens, right? So that makes sense. And so NLRs is a relatively large plant family. It is present not only in plants, but also in mammalian innate immunity. And what you can see from this figure there is a quite diverse numbers of NLRs in both plants and animals. We have pipelines and we have ways of rapidly identifying NLRs in genomes, and we use that based on the central NBR nucleotide binding domain that's typical to all of the NLRs. And when we talk about the NLRs and we think about the functional immunity, we want to look at the NLR diversity, right? And again, I want to quickly summarize to you what's known to contribute to NLR diversity across species. And I'll tell you today two stories about these two different aspects. One is the copy number variation. As you've seen, NLRs can vary greatly in the copy number, and that's both due to tandem duplications in the genomes, but also presence-absence variation. Then there's allelic variation, of course, point mutation, but also gene conversions. And what we've added to the that scheme is gene fusion, so NLRs can fuse with other plant genes. And then there's epigenetic and post-transcriptional variation of NLRs with alternative splicing and small RNA-based regulation. So today I'm going to tell you two stories. One is related to the copy number variation in plant genome, and another to this gene fusions. So this is my first graduate student, Erin. And Erin is absolutely fantastic. She came to the lab back in Earlham with no bioinformatics experience, and she wanted to do a PhD in genomics and bioinformatics and studying NLR evolution. And the very first thing that she picked up within probably the first 2 months is how to annotate those NLRs and look at the copy number variation of the NLRs across genomes. So she got a hold of the scripts and she analyzed every plant genome she could get her hands on. And she made this graph where we looked at the total number of NLRs in the species relative to the total number of proteins, with the idea being like, if what we see in the variation of number of NLRs is just reflection of expansion of the proteome, you know, we should see a trend. And we do see that. And then she was really interested to see, is there any type of correlation with NLR number and lifestyles of plants? So she's done a few different analyses, and she came up upon a very interesting phenomenon, which again started as a side project, and that's kind of the first paper she's publishing. What she found is there was a striking correlation in NLR copy number between all terrestrial plants and some of the aquatic plants that in both monocot and dicot lineages, they moved back to the water. So it's a secondary aquatic lifestyle. And when she looked at the evolution of the NLRs in both monocots and dicots, only including really high quality reference genomes and asking the question what happened in this aquatic species, in both monocots, she showed a great reduction of the NLRs, and in dicots that was even more pronounced. So what looked to us is that with moving to the aquatic environment, those plants lost their NLRs, while their very close relatives had much greater NLR number. And the question was, well, what type of NLRs did they lose? Did they lose all of them? They've retained some, you know, what's going on? So she did a tree. So this is a tree based on that common central NBARC domain, and included some reference species together with the aquatic species. And what we could quickly see is that those aquatic species lost most of their NLR clades and retained only a few, not the same ones in each aquatic species. And then there was an expansion, so those aquatics in blue, of those clades. So while most of those NLR have been lost, in particular, what was interesting to us is the dicot species, they lost a complete branch of TIR and the SLLRs, where the TIR is a signaling domain on the N-terminus. We know that in monocots, those proteins are actually absent most of the time, but then in dicot, it was striking that those particular clades were lost. So the next question Erin asked was, well, if they lost the NLRs, did they also lose the signaling components downstream of them? Or did they retain them, right? And so she started analyzing all known Arabidopsis signaling component and grouping them either in those effector targets, so proteins target that we know are targeted by effectors, or this is the signaling components downstream of the analogs, and this is a hormone assay, hormone signaling even downstream of that. And what we realized is indeed there was a group of genes downstream of analogs that's been convergently lost in both clades of aquatic species. And those group of genes include genes called EDS1, PAD4, and SAG101. And the striking, what was striking for us, we know that those are the proteins that form a protein complex. So it's a multiple components of a signaling protein complex that have been convergently lost. And B, we know that those are major signaling component downstream of those TEER and BS LLRs. And we've done a lot of analysis looking at annotation, assembly quality, RNA-seq, just trying to see maybe those genes have been missed, but they're just nowhere to be found in those species. So this is kind of Aaron's overview of this gene. So this, what we basically found that in addition to losing the receptors, these genes lost this downstream components, but retain whatever is downstream there, right? Remember, they didn't lose all of the receptors, and they retained some of the more downstream components. That was exciting. That makes sense. So the next question Aaron asked, and I thought that was quite clever, is if we look at all genes that are convergently lost in aquatic environment, what are any other genes that we might find? Because I told you, there's a big black box here. We still don't know exactly how that signaling happens and whether we have all of these components, right? So we decided to use those species to be able to say, can we do this genomic screen to ask if we can find some other components lost in this pathway? And what we call them angiosperm terrestrially retained, because that was a criteria for us. They had to be retained on species on land, and aquatic lost, or astral genes. And I'm going to outline two different methods, how we use it. One of them is called ortho-MCL. It's a very canonical method applied in comparative genomics, where you can have proteomes from different protein-encoding genes from different plant species. And what ortho-MCL does, it builds protein families from those combined proteomes to group them in the families. And we then asked, we wrote custom scripts to ask, what are the protein families that we see that are present in all terrestrial species and lost in all aquatic? And for the sake of computational resources, we did it separately for monocots and dicots, and then identified orthologous genes against each other. And this is the results. If we just look at the dicots, there's quite a number of genes that have been lost. If we look at the monocots, there's another clade, but the overlap in this, convergent loss, it's actually quite small. It was surprising to us. So I just told you about that. And the second method, we were, we were happy, but we were also not happy because it's just one computational method. And the big dependency there in ortho-MCL is how it calls whether something is a one gene family or not, right? And what we knew is that ortho-MCL it cuts gene families quite conservatively. It can split sometimes proteins from different gene families. So we adopted a second approach. It's called gene-seq2 family, which has a very similar approach of grouping the proteomes into the ortho groups, but it does slightly different algorithm behind it. If you're interested, that algorithm is published and there's a Galaxy workflow. And if you've ever worked with the plant ensemble and their orthology gene trees, the algorithm behind this gene-seq2 family is adapted from Plant Ensemble Compara database. And here we were able to group all the proteins together, and we had another group. what we know is that that genes stick to family. It tends to be on the opposite. It tends not to split genes into the kind of a smaller family, but group them together. So at the end, if we overlay those two analyses, we have our ortho-MCL done in a dicot species, monocot species. We have both of them in G62 family. There are only 5 genes that no matter how we cut these genomes, how we look at them, they're present. And there are overlaps that I told you here between ortho-MCL, monocot, dicot, and these genes are called slightly differently. Among the 5 genes that we found are this is now, unbiased. This is our EDS PAD4, and a few other genes such as NRG8DR1 that since we've done the analysis, they've been shown to be other NLRs that are required for the receptors. And among others, what we decided to focus on, we decided to say, well, we'll take everything that both the ortho-MCL code, and we'll take everything that genes stick to family code, and we'll take that overlap and try to look what's there. Because for us, that's very exciting. There might be like new candidates in plant immunity there. And the next question that we asked was, what's their expression profiles? Can we also sort these genes based on the known expression profiles? Are any of them induced during immunity? Similar, so we know EDS1 is induced during immunity. And maybe we were always concerned thinking, maybe there are other pathways lost in aquatic plants. So if we again look at all the expression data sets that are out there, and we took all the expression data sets for about 140 data sets that include microarrays and RNA-seq for Arabidopsis, and slightly smaller number, but still a big number for rice. And we look, what are the conditions under which these genes are, groups of these genes, significant groups of these genes are differentially expressed. And we did, basically there are only 2 conditions that came up. One was biotic stress. We were happy, kind of we are digging in the right place. And there were a significant group of those astral genes that were differentially expressed during P. syringae and Magna Portha arising in the rice. And in mutants of this is a SA signaling, in different mutants, they were also affected. And interestingly, there was only one other condition that showed differential expression of the astral genes. And there was drought. And for us, it was kind of an aha moment. The aquatic plants lost some of the components of the drought response. But it was also nice to see that we might be predicting not just the immunity, but we're doing this analysis to predict something else. So to summarize this type of analysis, what I told you is we use comparative genomics to identify known components that are lost in plants that change their habitat to aquatic from terrestrial, and they're retained in the terrestrial. And we have putative placement of some of these known genes, but also some of the other genes here in the pathway. And also there are candidates that we can place in some of the drought responses. What is striking to me, and this is my last slide on this part, is not just that aquatic plants lost. If they would have lost completely all of their NLRs and all of this pathway, I would have been, okay, fine. But what's striking is that they retained some of the NLRs, right? And they retained some of the component signaling. So what means that they rewired their immune response to adapt to that aquatic environment. Similar with the drought, it's not like they just wiped out, we don't need the drought anymore. We can actually, you could drought stress a duckweed or an aquatic plant, but it looks like they lost some of the major regulators again of this response. That means they probably rewired the response that was left. So the second quick project that I want to show you, and all of the data, most of the data, over 50% of the data I'm showing today is unpublished or it's on bioRxiv, is we noticed another outlier in this case, and it's maize. So maize has a really well-sequenced genome, absolutely great annotation, but it has a low number of NLRs compared to its sister species. Again, we decided to understand what was going on there. And first thing we've done, again, it's a phylogeny. And here's, I can show you the different scenario, contrasting scenario of a phylogeny of, again, NLRs on this central NBR domain between sorghum and maize, so sorghum very closely related species. And what you see that maize pruned its NLRs across the tree and retained one member from almost every clade, but it's greatly reduced the number. And we know that it's not that, let's say, sorghum expanded this NLRs. We could look at more of ancestral species. It's indeed that maize somehow lost them. Now, the sequence genomes now, and what I talk about here is elite maize varieties that underwent extensive inbreeding and domestication. So the project that we started, and we reached out to the maize community, and the maize community is absolutely great. They sent us seeds, they sent us samples, they sent us unpublished genomes. We started to address this question, well, this reduction could be due to inbreeding or domestication. And for that, we picked samples that represented elite maize lines, and we know the number there, or maize land races. And in Teosinte, we were able to pick outcrossing and inbred lines, and that would address the inbreeding hypothesis, and as well as domesticated, non-domesticated relatives. And we designed a sequence capture for NLRs. In the capture world, sequencing NLR specifically is called RenSeq, but it's essentially just capturing that family with specialized baits designed to hybridize to the sequence and pull them down for sequencing. And we now sequence about 80 members across this clade, looking at the most diverse species with PAG-bio. So we have PAG-bio long reads that we're able to address us this question. So that's kind of giving you a flavor what can happen to NLRs, very different scenarios and different lineages. And for the, how much time do we have?
15 more minutes.
15 more minutes, okay. For the rest of the talk, I want to quickly focus on gene fusions and want to bring these two examples, RS1, RPS4, and RGAF5, RGA4. There's two known NLRs that are functional, and what's been unusual about them, they have those extended non-canonical domains. They've been known for a while, and until their functional analysis shown the function of this domain, they've kind of forgotten. And what these domains do, they serve as baits for the pathogen effector molecules. They resemble those proteins that are targeted by the effectors, and they serve as integrated decoys into the NLRs that monitor the effectors. The question we ask is, if we can look at all of the NLRs, can we look at this is phenomenon that's been described for two genes as widespread across the plants, and how many more fusions we can find? And so long story short, we did find that about 10% of these NLRs, they have fusions to other plant genes. That's a widespread phenomenon, and those are quite diverse fusions. Since then, many other, and we term those NLRs with integrated domains, since then, several other NLRs have been recognized. And what are those integrated domains tell us, right? This is a genomic screen that could identify proteins that might be targeted by the effectors, right? So we could, by the looking at just identity of this fused baits, we can tell what are the other proteins might be important for plant pathogen interactions. So we did a KEGG analysis looking at the homologs of those integrations. And we found, of course, plant pathogen interaction MAP kinases on top of it. But where we also found is quite a number of metabolic pathways, and I talked a little bit about that yesterday, that are enriched in those integrations. Meaning this is a plant cellular pathways monitored by plant immunity and likely modified by the pathogens. So how can NLRs tolerate these fusions? We had two hypotheses. One is that NLR IDs form at random during NLR evolution and is followed by adaptation. And the second is that there are evolutionary hotspots in this diversity. There's a specific place where those fusions happen more often. And to long story short, we made a tree. We mapped on the tree where, and this is 9 grass genomes, where those NLR IDs were. And we found that were several clades that were highly enriched in those fusions. And so that's been published, so I'm gonna go quickly. And again, there's one clade in which there was more than like 60% NLRs fused to very diverse domains. We looked in wheat because it's a polyploid, and we looked at different subgenomes. And we see that even in the subgenomes of wheat, there are different fusions even to the orthologous homologous sequences. So that's, again, what's shown here. There could be 2 closely related homologs, and they can have different domain attachments. So this is an ongoing evolutionary process. And we've spent quite a lot of time now looking at the mechanisms of how that happens. We were able to tell that it's an interchromosomal transfer of genes, and it's a copy and paste process. So what we, the model how we think evolutionary, what happens is there has been an adaptation of particular NLR to accept these new fusions. And together with the Matthew Moscow lab, we were able to identify a protein motif that was specific to this clade that we now think might facilitate regulation of the NLRs by these fusions. And interestingly, on a structure, that protein motif maps to the last hydrophobic cap of the LLR that kind of closes that LLR surface. Now, can you also see from the slides that those NLRs often function in pairs, so they often require another NLR. So now the question we asked, if this is NLR that has an integrated bait attached to it, and the known ones have a helper NLR that often genetically linked. So it's a perfect, basically, genetic stack where the 2 genes are correlated together. We asked whether this is true, again, on the genome-wide level. And we've designed an algorithm to look for those head-to-head pairs and looked for, across any NLRs, what type of paired relationship we have. And this is NLRs belonging to different clades. What we saw that our clade of interest with integration, 16, often paired with another clade, clade 7. And this is just a circus plot, if you imagine a phylogeny in between, that displays all of this paired relationship, this MIC1, clade 16, clade 7. So we now postulate that C7, that clade 7, is this helper clade, while this MIC1 is the sensor clade that baits for the pathogen. And what I want to bring to your attention as well is I showed you those 2 cases of Arabidopsis and rice where there was this paired relationship, helper-sensor relationship. And if we look in the context of the evolution of NLRs across monocot and dicots, this is essentially a convergent evolution. Those helper-sensor pairs that are in head-to-head adaptation in Arabidopsis and rice, they evolved independently in these 2 clades. How? We don't know. Why head-to-head? For sure, we don't know. And another helper clade here that's important for solanaceous that's been studied for a while. It's a different clade. So that's, again, like an example of convergent evolution. Thinking about the mechanism so far, I wish I could tell you the exact mechanism, but so far we've excluded retrotrans positions because these fusions still have introns. And we don't see any transposon signature associated with it, and we've been looking so far. Okay, a couple more minutes, like thinking, yeah, 5, 10 minutes. We've started looking within species. So now I told you, okay, there's great amount of diversity happens across species. So far, I've already told you across species. We've been starting to look within species, and specifically looking at those NLR IDs, highly evolving clades. This is 13 rice species. This is published now by some of our collaborators, Josh Chan and Doreen Ware. If you look at the loci, this is scrambled eggs. Those loci that harbor those NLR IDs, they evolve so rapidly. Now again, this is closely related, slightly different species. At that time, Brachypodium pie genome came out. There was no NLR analysis, so we decided to look. And we first saw that number of NLRs varied maybe 2-fold within the species. The number of NLR IDs was there, Most of the NLRIDs were newly formed. They were present only in one genome, but not any others. Some were segregating. They were present in half of the genomes, but not in other half of the genomes, and some of them were fixed. So we'll look at those fixed, and we looked what's happening to them. And this is showing you a synteny between brachy and rice for that locus. You know, it's a highly divergent locus in rice. It's present in brachy. There's another duplication. And this is a schematic, like this is my last set of slides where I want to show you within a brachy species how much evolution happening in those NLR loci. This is the reference species, right? If we look just in the reference, and this is the NLR ID here in the purple, there are some synthetic genes around it. There are a lot of duplications that happen to this gene. And moreover, in many strains, there are gene size insertions that just come in of this locus. And these genes that come into this locus, if we look at them closely, they resemble integrated domains. So we think those are the donors, the donor material that can get integrated. So this is much bigger kind of genomic question now that we are working on. So with that, I want to summarize what I just told you. NLRs is an amazing plant family. NLR repertoires vary greatly. In aquatic plants, there's low, but if we look at crops, as low as 30 in cucumber to over 2,000 in wheat. Maize has a very unusually low number in NLR that we're trying to understand why. And aquatic plants are convergently lost, both NLRs and downstream signaling components. And then those NLRs, they also are good markers for understanding how the plant proteins can fuse with each other and function. And also they allow us to dissect plant-microbe interaction, revealing what might be the proteins targeted by the pathogen. But then I want to thank my group, especially Erin and Andrew, who moved with me from Norwich to Berkeley, as well as all of the lab alumni and our collaborators. Thanks.
So I think we have time for some questions, and I just asked that for people online or in the recording, can you just repeat the question in the microphone? So are there any questions from grad students or postdocs in Ithaca? Okay, yeah, go ahead.
Aquatic plants have fewer AR genes than terrestrial species. What do you think is the evolutionary driver?
Okay, that's a really good question. We don't know the exact answer, but we can think of it two different ways. Either those NLRs are not needed in aquatic environments, so they present a cost that can be lost, right? Or they're detrimental, or some of the signaling pathways or some of the responses are detrimental. We don't yet know how to distinguish it besides maybe transforming duckweed to see if we put them back, like, does it really kill it?
Adam? Former graduate student.
Right, yeah, we're all floors open, yeah.
A follow-up question on Lionel's question. That is, did you ever sort of flip your analysis and ask whether there are innovations in aquatic species that are absent from terrestrial plants? Sort of as a corollary to that, specifically for the drought tolerance genes in terrestrial plants, do you think their absence is simply in aquatic plants is shedding baggage, or do you think that those loss of function changes are essential as an essential step for adaptation to?
So the question for the remote audience is whether the aquatic plants compensated by, as I understood the question, by expanding other protein families, for example, or relying on other pathways more heavily. And the second part of the question was whether that loss, especially in a drought, that was the detriment, because it was detrimental, right?
Yes.
Mm-hmm.
So losing it was essential.
Whether losing it was essential or it was just a passive kind of loss of the genes. Now,
the first question too, specifically what I'm thinking about, are aquatic plants a potential source of innovation?
Yeah, aquatic plants, yeah.
Or drought tolerance?
Are aquatic plants source of innovation for drought tolerance? That's an excellent question. I have a DOE pre-proposal submitted on that, and I have a duckweed collaborator at JBay who's grew a duckweed and drought stressed it already and ready to extract RNA from it. So it's an unusual direction for my lab, but I'm happy to take it if we get the funding. I do think that it is duckweed. It is a sort of innovation. I think they rewired because the downstream, some of the downstream drought response genes, they're there. It's just those master regulators are not. And that's the same with, let's say, the immunity, the SA pathway is there, the NPR pathway is there, but this kind of the middle layer and some of the upstream components are not. So I do think it's a case of rewiring, and I think it might be a source of novelty and adaptation.
So I might have missed this during the ringing because I got a little distracted, but there were clades that are hotspots for IDs going in. Are there like genomic loci, like physical locations? And then is that synthetic across like closely related species, or do those move around as well?
Yes, so the question is whether there are hotspots, clades for fusions, and the answer is in cereals and monocots, yes. There is that hotspot clade. In, let's say, Arabidopsis, there isn't, that we found. And the next question is whether there are genomic locations. And we do think that the genome architecture and the surrounding loci do play a role. But what we think it's a several loci distributed across the genome. So it wouldn't be just like one locus. And if we look at, let's say, brachyrice, that is conserved, that the presence of the NLRIDs and that diversity is in the synthetic locus. So I've kind of skipped that probably fast. But that's an excellent question.
So any questions online, feel free to hop in. Just give them a second. And if not, Jinhua, it looked like you had a question.
So the loss of the EDS platform and then the loss of NLR, have you looked at which one happens first?
Loss of EDS1 PAT4 and loss of NLRs, which one happened first? We don't know because there's not enough resolution in the genomic sequences of the kind of the relatives. And there's nothing remaining of those signaling component genes. Overall, aquatic plants do undergo genome reduction. And it's just those are the genes that are convergently lost in all of the species. What we could tell is, what we can tell in one of the aquatic plants, which has a remaining, let's say, one NLR, that NLR is going out. So, and some of the NLRs we see as well, what happens basically, it did lose the DS1 PAT4, and we still see some of the NLRs just kind of on their way out. They would lose the motifs, they would become pseudogenes in the sequences.
I think we have time for one more.
Yeah, for the cereals, you have like two outliers. You have the maize that have really low copies, and you think domestication may play a role in that. But on the other hand, like you have wheat that has really extremely high copy numbers. So why do you?
Yeah. So we have designed RenSeq on wheat, and we're sequencing about 60 lines as well, and our hypothesis there, what we want to distinguish is hybridization, polyploidy, from, again, domestication, other events. So we're sequencing a panel of wild tetraploid wheat called wild Emma, and comparing it to the domesticated durum wheat to make that distinction. But you're absolutely right, something unusual on the opposite spectrum happened in wheat, where there's an expansion of NLRs, and it's not just a sum of the diploid progenitors. Diploid wheats have about 400, 500 NLRs, times 3 in the hexaploid, that should be 1,500. What we see is close to 3,000.
So just a reminder that if you're a grad student or postdoc and interested in joining us for lunch, that'll be coming up right now. And please join me once again in thanking Ksenia.
This has been a production of Cornell University, on the web at cornell.edu.